Transthyretin. What is it? Transthyretin, also referred to as TTR, is a human protein secreted by the liver that helps with transport of retinol and the hormone thyroxine in the body. In its natural state, transthyretin functions as a tetramer, meaning that four identical transthyretin proteins are working closely together. This tetramer is not permanently held together, though, and the proteins can separate into dimers or monomers. When acting as a monomer, transthyretin is unstable and has the potential to misfold. Should enough monomers misfold, amyloid is formed as a tight aggregation of multiple misfolded beta sheets. Transthyretin amyloidosis, ATTR, is a disease associated with these amyloids. Symptoms range from loss of sensation in limbs to cardiac arrhythmias. The age of onset can range from 20 to over 100. There are two distinct categories of ATTR, being Familial Amyloid Polyneuropathy, FAP, and Senile Systemic Amyloidosis, SSA. SSA occurs in the wild-type gene for transthyretin and has a much later age of onset, generally after 70 years. FAP occurs in mutated forms of the transthyretin gene. Over 100 mutations have been identified. Each mutation expresses differently, with some lying dormant and others causing earlier ages of onset with more aggressive symptoms. Though many studies have taken place studying these mutations in families and communities across the globe, it is not currently known how these mutations are differing so dramatically. So what am I doing? This summer, I will be modeling 3 selected mutations in 2 yeast strains to look at cell viability in these various strain mutation combinations. I will be using Saccharomyces cerevisiae, yeast, as my model organism due to their ability to quickly reproduce and easily take up outside genes on plasmids. This will allow me to transform the human gene into live yeast cells to study the mutations in vivo. I started the summer with the wild-type transthyretin gene on a plasmid flanked by a GPD promoter and CFP fluorescent. I will be using the gateway cloning system to clone the transthyretin gene into a plasmid flanked by a GAL promoter and GFP fluorescent while performing the mutations along the way. First, in the gateway system, the transthyretin gene will be removed from its current plasmid and have attachment sites added to either end. Through the BP recombination reaction, the transthyretin gene will be recombined into an entry vector. At this point, mutagenesis will take place to create each of the 3-point mutations to be studied. Then, through the LR recombination reaction, each of the genes will be cloned into the destination vector containing the GAL promoter and GFP fluorescent sites. After these mutations have been constructed, I will then transform each of the plasmid products into either of the 2 yeast strains, one being more prone to amyloid formation. After this transformation, I will monitor the yeast cells' viability by looking to see if they are surviving, thriving, and multiplying. And that is my 2016 Summer Research Program project.